Viruses are tricky. They use all sorts of unusual mechanisms during their attacks on cells. HIV is no exception. It has a retrovirus, which means that it has the ability to insert its genetic material into the genome of the cells that it infects. But infectious HIV particles carry their genome in RNA strands. Somehow during infection, the virus needs to make a DNA copy of its RNA genome. This is very unusual because all of the normal cellular machinery is designed to make RNA copies from DNA, but not the reverse. DNA is normally only created using other DNA strands as a template. This tricky reversal of the synthesis is performed by the enzyme reverse transcriptase, shown here from PDP entry. Inside its large claw-shaped active site, it copies the HIV RNA and creates a double-stranded DNA version of the viral genome. This then integrates into the cell's DNA and later instructs the cell to make additional copies of the virus.
Viruses are tiny. They only carry enough genetic material to encode a few proteins. Many viruses, such as poliovirus and rhinovirus, carry the bare minimum, just enough to specify their own structure and get synthesis started once they get inside cells. The genome of HIV, on the other hand, carries instructions for building a few enzymes that are used in the reproduction of the virus. Reverse transcriptase is one of these enzymes, but space in the HIV genome is still at a premium, so reverse transcriptase is encoded in a tricky way. It is composed of two different subunits, both are encoded by the same gene. After the protein is made, one of the subunits is clipped to a smaller size so that it can form the proper mate with one full-size subunit.
Reverse transcriptase performs a remarkable feat, reversing the normal flow of genetic information, but it is rather sloppy in its job. The polymerases used to make DNA and RNA cells are very accurate and make very few mistakes. This is essential because they are the caretakers of our genetic information, and mistakes may be passed on to our offspring. Reverse transcriptase, on the other hand, makes a lot of mistakes, up to about 1 in every 2,000 bases that it copies. You might think that this would cause severe problems, but in fact, this high error rate turns out to be an advantage for the virus in this era of drug treatment. The error allows HIV to mutate rapidly, finding drug-resistant strains in a matter of weeks after a treatment begins. Fortunately, the recent development of treatments that combine several drugs are often effective.
The current collection of highly effective drugs for fighting HIV infection are a major success of modern drug design. Two types of drugs are used for blocking the action of reverse transcriptase and stopping HIV infection. One type is a modified nucleotide with missing connectors, such as the drug AZT. These are used by the enzyme like normal nucleotides and added to the growing chain, but since they are missing a site for connecting the next nucleotide, the synthesis of the DNA chain is stopped. The other type of drug binds on the backside of the enzyme and changes the shape of the active site, blocking its action. The drug nevirapine, shows here in white from DPD entry, is an example of this type of drug. Notice how it is located just under the floor of the active site below the big groove that binds to DNA and RNA.